So you could imagine extracting the DNA from such a fossil, making sequencing libraries, just sequencing everything that's in there, and in the computer identify what is human-like there. And there was a great effort to look through very many different sites to find good specimens that would contain enough endogenous DNA to do this. It was particularly one site in Croatia, Vindija Cave, and this specimen that turned out to be very useful, containing up to 2 or 3% endogenous Neanderthal DNA. There was also a lot of improvement of technology at the time, where we got much, much better in extracting short fragments of DNA and turning them into libraries. Sequencing technology got better. And indeed, there's sort of the size distribution, the size distribution is indeed very small in these things. And when we then started sequencing this fragment, one interesting thing turned up towards the end of the molecules, particularly, where when one then looked at the substitution patterns, you found that C's, cytosines, at the end of the molecules very often appeared mutated to T. And that turned out to be the result of the most common form of modification in ancient DNA, which is hydrolytic deamination of cytosine to uracils, which are then by polymerases read as T. So this is then a problem. You have to take it into account when you align your sequences to present-day genomes, but it's also an advantage because then use it as a marker for molecules that are truly ancient, because it takes hundreds and thousands of years for this to accumulate at a reasonable frequency. So we used 3 bones from that site, sequenced a lot of molecules from them, aligned them to the human genome, and much of the bioinformatics, both at the time and currently in our work, is led by Janet Kelso and her group. To sort of align these to the human genome, taking particularly the C to T substitutions towards the end of the molecules into account. And we then, in 2010, as we heard, had a first coarse draft version of the genome, where a little over half of the alignable genome was available. So a big question then in the beginning was, how were Neanderthals related to present-day people? So an expectation, if Neanderthals had contributed to present-day Europeans, would be that Europeans today should share more genetic variants with Neanderthals than Africans share with Neanderthals. As Neanderthals had never existed in Africa, there was no reason to assume that they would have contributed to Africans. So to address that and analyze other aspects of the genome, we put together a big consortium. An important person in that worked in Leipzig at the time was Ed Green. There was also David Reich at the Broad Institute and Monty Slatkin at Berkeley that were very influential in this. And one analysis then was to compare the Neanderthal genome here to genomes we sequenced to have similar patterns of errors in our sequences from different parts of the world. So you could then do a very simple analysis where you simply identify differences between 2 present-day genomes, to start with 2 Africans and compare that to the Neanderthal and say, how often does a Neanderthal match that African or that African? And you will expect that to be 50-50, since there's no reason to assume that a Neanderthal would be more closely related to one African than another, and indeed that turned out to be the case. If you then take a European individual and an African individual and do the same analysis, we found statistically significantly more matching to the European individual. Another example I want to bring then has to do with reproductive biology. It has to do with progesterone, that you know is a hormone that has many functions, but one major function is that it's produced by the corpus luteum after ovulation and prepares endometrium for a possible pregnancy. And what we're interested in here is the receptor, the progesterone receptor. It was already known that there is a variant of the progesterone receptor that exists that's associated with preterm births, so premature babies. What is striking is then that this variant exists hardly at all in Africa, but do occur in populations outside Africa. Wherever you see this distribution, it's very likely that it actually comes from Neanderthals. And when people noticed this, it was then even suggested that this has to maybe a selective disadvantage of Neanderthals to have premature babies that are, of course, at risk to die. However, what is also becoming possible now is that we have thousands and now over 10,000 genomes of present-day of modern humans over the last 20,000 years. So we begin to be able to follow the frequency of variants across time by looking in present-day human genomes. So if you have this little movie here where we just have the archaic genomes in red, we have carriers of this risk variant for preterm births are black, other ones are gray, and we walk forward in time from 15,000 years ago towards present day. Around 7,000 years ago, you will see how this variant almost explodes in frequency, which makes no sense if it is a risk factor for premature birth, preterm births, right? So that then caused us to look again in the UK Biobank. And if we look at associations there now with the modern allele, the non-Neanderthal variant, what we find is that it is significantly associated with bleeding early in pregnancy, with miscarriages, and it is negatively associated with numbers of full sisters and actually brothers, but not significantly so. So this is then quite surprising. The non-Neanderthal version is actually protective, is actually a risk factor for miscarriages. So it turns out then that the Neanderthal version here is indeed associated with preterm births, but it's also protective against miscarriages and results in more live births. And we're beginning to understand the function of that also, in that if you look what carriers of this Neanderthal version express more of the receptor. So you can imagine that you have more of a progesterone effect if you have more of the receptor. And indeed, is sort of compatible with that is recent observations that if you give progesterone to women who have experienced several miscarriages, you can prevent or increase the likelihood of a live birth, compatible with that more progesterone effect, either by giving more progesterone or having more of the receptor may be good. And this is sort of a pattern that many genetic variants that are already known turn out to come from Neanderthals when we now have the Neanderthal version.